Yeah, so as far as I said, I'm Alessandro. I work at the Sainsbury Welcome Center, mostly on Brainglobe, and I will talk to you specifically today about how to align your electrophysiological data to histological data. First of all, why would we want to do this? This is probably kind of obvious for you guys, but you will be recording lots of cells at once, and you need to know where you're recording from, so which anatomical regions your recordings are from. And maybe you also want to combine multiple recordings from multiple animals into the same coordinate space to be able to compare them and do your analysis on them. So as you know, typically you would label your electrophysiological probe with something fluorescent like dye, and then you insert your probe, you do your experiment, and then after you finish your experiments, you take your mouse brain and you image it. And so this is the kind of data I'm talking about. To align to histology, you have this kind of whole brain microscopy data. This can be light sheet microscopy or 2-photon zero section or something like that. And then the next step is that to do this is you want to go from your raw data, where you can see your probe shining fluorescently, to segmenting your probe and knowing which atlas regions, which anatomical regions your probe is from. So you also need to align your data to an atlas. And this is the kind of information that you will end up with, where you see that these recording sites here are on the primary visual layer, and the ones lower down are in hippocampus, possibly from multiple animals. So you can have several probes from multiple animals in the same coordinate space. So how do you do this? I will have a demo later, but in principle, the Brainglobe ecosystem, there are 2 tools that allow you to do this that we'll go into more detail on. They're called BrainReg and Brainglobe segmentation. BrainReg is our registration tool to the atlas. So this allows you to align your whole brain imaging data to an atlas, and Brainglobe segmentation allows you to figure out where the probe is, and so then you can combine the 2. In the paper for this, we did some validation where we showed that the kind of anatomical probe, where the probe is anatomically, matches the electrophysiological signal. So here we have this typical signature, as far as I understand, in the primary visual cortex layer 5. It has this electrophysiological signature, and if we looked at primary cortex layer 5 in the anatomical data, we get differences of about 60-70 microns, depending on which person drew the probe manually. So this is something you can do for validation, but you also need to do this validation on your own data. Just this can give you kind of an expectation of how precise these methods are. So now here we have a quick demo. I will talk over it. The video is slightly out of date, but it should give you, the tools haven't changed that much. So first of all, you would open your raw data. You would go through it and check where your probe is. So the first step is to align to an atlas. We have all of our tools have this graphical user interface in Apari, which you can see here. And you can now see the probe light up on the top left. And then you would open our BrainReg plugin. You would say what atlas you want to align to. And you shouldn't need to, especially for mouse data, you shouldn't need to change the parameters that much. You would just input kind of which way around your mouse brain is oriented in, and the voxel size that you imaged with. And then you hit run. And this allows, this usually takes a couple of minutes, so this is a bit quicker in the video, just through editing magic, but this would take a couple of minutes to maybe like up to an hour for very high resolution data. And this allows you to overlay, to calculate the transformation between the atlas and your data, and to overlay the regions. So this will save all this information into a project folder. that you can then use to segment your probe. So then to segment your probe, you would open a different plugin called Brainglobe segmentation, and you can load your project folder that you got from the previous step. And here we're loading the project in atlas space rather than sample space, so the data will look slightly differently because it's now aligned, it's now in the atlas coordinate space, so it's more symmetrical here, but it is the same data. Again, you can see the atlas regions. We have several types of segmentation. On the top right here, you can see track tracing or region segmentation, because we have a one-dimensional track here, so we would use the track tracing, and we would draw roughly equidistant points in the middle of the track along it from the surface to the inside of the brain. So you can then maybe double check your points and then hit trace tracks. And there's a point here called spline points. So this is the number of points along the track that this new layer that is highlighted in blue on the left will have. And so it's probably wise to choose like either the number of recording sites that you have along this shank, or some kind of physical distance. So if you know your track is, I don't know, a millimeter inside, and you use 1,000 points, then you know every measurement you take is at 1 micron, so 1,000ths of a millimeter along the track. And then you get output like this, where you, at each point of those 1,000 points along the track, you get a region ID that you can feed into your further analysis. And so you can do this for Neuropixels probes, but also for other electrophysiological probes, or to segment viral injections. So here you would measure more like bulk fluorescence or optical fibers to determine which anatomical structures they belong to. As I said, this is part of the Brainglobe ecosystem. One of the central parts of the Brainglobe ecosystem is also the Atlas API, so that means that you can use these tools with any available atlas. So typically it's the Allen Mouse Atlas. I think many people use that. But there's a number of atlases that are available, including other mouse atlases. So there's the Enhanced and Unified Mouse Atlas. We have some developmental mouse atlases, or if you study more esoteric species, also we have, I don't know, a blind Mexican cavefish brain atlas. There's a few more, not all of them are here. So if you go to our website, there's more atlases. We recently added another developmental mouse atlas, the Demba developmental mouse atlas. If any of you are working on that kind of application. Finally, this is a community project, so we have to thank a lot of people for, as well as the funders, a lot of people for support. So we have over 100 people that have contributed to the Brainglobe code base, and so you are also welcome to get involved with that. I will leave you with a few resources. So we have a website where you can find tutorials, including silicon probe tracking tutorials, which guides you to the demo that I just sort of showed you in the video on how to do that. All our code is on GitHub. If you would like help with any of the tools, you can go to the ImageSC forum and tag posts with the Brainglobe tag, and we will get notified and answer. If you want to discuss more software development aspects, you can also join our developer chat. So that should give you a short overview of probe tracking in Brainglobe, and I'm happy to answer questions.